have a presentation today from some scientists, a person who has a new device, and actually the business end as well. I'm Nurse Linda. Many of you know me. You've been to some of our webinars before, so welcome, and welcome to new people. I will describe myself for you. I have fair skin, which is naturally ruddy, so if I look a little pink, that's the way I look. I wear glasses, and I have mostly brown hair. It looks brown on the camera, but there are little grays slipping in there. And there's a white background behind me. Today, I would like to welcome Dr. Reggie Edgerton, Mr. Ignacio Montoya, and Mr. Craig Gadd, who are here to talk about a new device that they have with some transcutaneous spinal neuromodulation. So welcome, gentlemen. And we'll start off with Dr. Edgerton. If you could tell us a little bit about how you got into this game and what entices you with neuromodulation of the spine.
Even when I was growing up, I was interested in how we control movement, particularly because I was interested in sports. And so eventually I realized that there really was a lot that needed to be learned about how do we control movement. The science was not very well advanced in this area. So I decided to study the basic science of how we control movement. And my first entry was focusing on muscle, and then eventually I migrated to the spinal cord. And now I'm even forced to consider that the brain is important. Anyway, I think one of the things that I think is an important point is that I was not trained as a neurologist, and therefore I did not have any preconceived notions about what was not possible and what was possible. And I was just focused on the science. And basically what has happened is the science has led me exactly where we are today. If you follow the science, each experiment over a period of years, many years and decades, each result led to what the next obvious question was. And the central question that became that I developed with colleagues early on was the fact that the spinal cord was able to do some remarkable things even without input from the brain. And as a result of that, I started asking the question, and I'm still asking the same question, and that is, to what extent after paralysis can one take advantage of the function that is left after a spinal cord injury? What is the residual physiological organization of our nervous system that helps to control movement? To what degree can it operate relatively independent of the brain? And of course, that leads into the concept of the spinal cord circuitry being smart. And we're still finding out that it keeps getting smarter and smarter, or we are getting smarter and learning how smart the spinal cord really is. And basically that translates into how much we do in our movements are performed relatively automatic and not just simply thinking of all the details. So the science has led me here, and the things that we have learned, along with many other labs, you put all those things together, and you kind of end up where we are. But when I thought of the original question of to what degree can we take advantage of the residual information, I still cling to that and really see that actually what happened at that time, which was in the mid-70s, was that the seed was planted. And everyone thinks of how much has been accomplished today. And the way I see it, it has taken decades for that seed to develop into a tree that is providing lots of fruit for us to pick.
Well, the nervous system is very complex, and we certainly are learning more and more about it every single day. So what is your current project that you're working with transcutaneous spinal stimulation?
Well, all of the experiments now are related to how we can change the properties of the spinal cord, the neuronal networks within the spinal cord and the brain, so it will respond in a more highly functional way. In other words, what we tell the nervous system to do is only part of the problem. The other problem is the physiological state of the spinal cord, and that defines how we respond to a given stimulus. And I think this aspect of the control has not been studied as seriously as the cortical control and driving the nervous system. We know, for example, routinely that our physiological state changes throughout every day. If we haven't eaten for a while, we become hungry. And then how our food tastes is a function of how hungry we are. And so the nervous system can change to the extent that in one case, a voluntary effort would cause you to perform one motion, but if it's in another physiological state, it could, with the same signal, it can do exactly the opposite. So we need to know a lot about the sensory system. And so our focus now is much more on how we can recover sensation, because it's been known for years you can't control something that you can't sense. You have to have some information about where your body is before the brain and spinal cord can perform a given task. So the sensory system and the regaining of sensation is I think is one of the newer things that we need to pay attention to. And it has turned out that almost all of the experiments, but this has been known for decades actually, but a major piece of sensory information is proprioception. And proprioception is a major tool that we can use to drive the spinal cord and the brain as much as the brain, the cortex, driving this sensory system. All of the sensory systems are important. And one more thing about this with respect to injury is our concept is we want to be able to take advantage of every source of sensory information that's left in order to help to tell the spinal cord what to do and how to do it.
That proprioception is really a key. I see that a lot with people trying to figure out their body in space, and then of course making your body work within that space. There was a question that did come through in the question box about synchronization of the spine. Could you talk a little bit about that?
Well, synchronization is a key. For example, there's only two things the nervous system has to do in order to determine what the movement's going to be. Number one, it has to activate each muscle, and that is each motor pool, all the motor neurons that go to that muscle. And that is the mechanisms for that's pretty well understood. But what is not understood and is a major puzzle is how do all these muscles become coordinated? And you can perform many movements with the same muscles being activated, but it's determined what the motor result is, the behavior is really determined by how they're coordinated. And another way of saying that is, how are these muscles synchronized? So that has to happen for locomotion, of course. Much of that control is from the spinal cord. So the coordination is a major thing. And now that we have found out repeatedly that individuals that have been categorized as Asia A and having no motor potential for movement, we know now that they can regain connectivity with the brain. So now, in fairly regular occasions, these individuals can regain some connectivity between the brain and the spinal cord. So now, what we have to understand, how does the brain and the spinal circuitry become synchronized? Because the spinal cord,
somebody. May I help you?
the spinal cord has to be told the same thing from the brain and the spinal cord spinal cord.
So how are you making that happen? What are you doing to make this happen?
Well, we're trying to take advantage of all of the detailed mechanistic things that have been learned over a long period of time. Now, what we know is, as I mentioned earlier, almost all features of the details of our movements are determined automatically. And this automaticity was demonstrated decades ago through a concept that's referred to as central pattern generation. And that is where you can take the neurons from the lumbosacral spinal cord and activate them biochemically or electrically, and it will generate a remarkably normal coordinated pattern. So that tells us immediately that there's a lot of automaticity that's going on to coordinate these motor pools so that it knows how to step and stand and so forth. So that is a basic biological concept that has been fundamental. So we have some ideas now of how this central pattern generator can now interact with the sensory information. So the central pattern generation capability can do nothing without proprioception. And when you give it proprioception, the spinal cord knows what to do with that specific pattern of proprioception. And that's a concept that we are excited about developing right now, because we think this is a key to understanding how to get these systems coordinated. But still, the issue that we don't know how, but we think it's a matter of training, is we were trying to train the brain as well as the spinal circuitry to focus on the same activity at the same time. And we're thinking that there's intrinsic guidance principles that is helping the brain to know how to make connections to the spinal cord and vice versa. So there's no question now that the use-dependent plasticity is very fundamental and is going to be a major factor in driving this synchronization.
If I may add there, when Reggie talks about the proprioception, the best way to understand it, or an analogy for it, is the nervous system, or the proprioception, is like a GPS system. If you want to go from point A to point B, that's only possible if you know where you are in space at point A. And that's what proprioception really tells you, is where are your limbs? What is the physiological state of that that network, for you to understand where you are at point A, and how do you then go from point A to point B. To go from point A to point B, there are probably a million different options and ways you can do that. And that's where the training, the neuromodulation, and the voluntary control comes into picture.
That's a fascinating concept. The way you explain that makes it very clear. If you think about your GPS system and all the different routes you can take, but the spinal cord and the brain are going to take the route that's the best route. So that's a good analogy. Thank you for sharing that. Could you talk a little bit more about that central pattern generator?
Well, I think I've made the major points. There's a lot of science that has gone on in the last decades to try to identify exactly what the central pattern generator is. Where is it and which neurons? Is there a specific type of neuron that's responsible for a given part of that action? And a lot of progress has been made. Now, our perspective is from I refer to it as a top-down perspective. And I basically think as a systems biologist. And one of the things that has become amazing, even though we've known this for many years, basic physiologists have known this, is how integrated all of our organ systems are. And so if you take a highly reduced preparation, a neuron, for example, there's just a limit to what a neuron can do. It can't in itself learn, it can't perform an action, it can't perform a movement. The more you integrate the system, the more you realize what the body can do. So if you're studying a system that is reduced to a significant degree, you're studying a system that can't even do what the whole organism can do. And so we are constantly reminding ourselves that we need to know these new things that can be performed when you have all the body systems connected. And if you disconnect some of those systems, then you're automatically limiting the potential for your interpretation of your results as to what is really possible in the whole integrated system. And this goes back to what we're seeing, and Parag mentioned earlier, is that you can't affect just one organ system. If one organ system is affected, every organ system is affected indirectly in one way or another at one time. So we can use that as an advantage as well as the disadvantage of all the organ systems being affected by spinal cord injury. But now we're learning through use-dependent activity, as soon as you get a person standing and getting proprioception added and using other sensory systems, these other organ systems come into play. So it's been impressive for not only us, other laboratories. We saw this in our first subject that was implanted epidurally at University of Louisville in collaboration with Dr. Harkema and Dr. Gerasimenko and our team, where in the first subject, sexual function improved, bladder function, sensation, temperature control, all of these things start to come into play when you put the whole organ systems together.
And movement and sensation certainly is a huge part of these. Now, when you were doing this, when you did this study in this particular case, did you use males and females?
Well, we started this with one subject. This was our plan. All of the evidence from decades of experiments with animals had told us it was time to do this experiment in human subjects. And then when the experts were brought together, there was general agreement that we knew enough to try this in human, but we tried one. And our plan was to do this in 5. We had an NIH grant to perform this in 5 experiments, in 5 individuals in sequence. And we learned from each individual. And so we basically found very similar results in each of the individuals. Each of them regained voluntary control of some kind of their lower limbs as a result of epidural stimulation and training.
And so where have you moved from that? Are you still doing more subjects, or are you moving onward from that study?
Well, after that, we had the idea, this basically, Dr. Gerasimenko, when we were working together, and we're still working together, he came in and said, why don't we try this new with this stimulator that was used decades ago with muscle stimulation? And that's when we started the non-invasive stimulation, not of the muscle, but of the spinal cord. So we place these electrodes over the spinal cord. And we are basically finding amazingly similar results with transcutaneous stimulation as epidural stimulation. So that is almost our total focus right now, because we think the transcutaneous stimulation is going to have a greater impact than actually the epidural stimulation. But at the same time, I'm not saying that from the idea that we should only be doing transcutaneous stimulation. I think all of these different strategies that are being used in neuromodulation, I would like to see them all fully developed as much as possible, because it's almost certain that one patient is, there's going to be one intervention that's better for a patient, another patient, another intervention. And so I hope that all of them will be developed. But our focus right now, because we're convinced of its high impact, is to focus on how to maximize the potential of neuromodulation non-invasively, transcutaneously. The other point let me make is that there are multiple ways to neuromodulate the system. Another one is pharmacological. And our animal experiments also showed that pharmacological neuromodulation of the spinal cord is extremely effective in modulating locomotion. And we have done very little of that. There's a lot to be learned with the pharmacological perspective. But so far, we've focused on the electrical neuromodulation. And on top of that, the method of providing electrical neuromodulation has been, in the last 8 or 9 years anyway, has been with the non-invasive transcutaneous stimulation.
Well, the pharmacological certainly has a lot of implications. We'll be excited to hear about how all that turns out. I know that's in a bit earlier stages, as you said, but it's interesting to see the evolution of the transcutaneous, starting out with applying it surface electrodes on arms and legs over muscles and nerves, and then now applying it directly to the spinal cord, either through implantation or, as you said, the transcutaneous. So it's kind of come a full circle almost with all of that and how that's working out. And you can see from your slide how many different body functions are improved by doing the spinal stimulation. And I know you have Ignacio there with you, who is using the spinal stimulation. So would you be willing to talk about what you have seen? What made you decide to do this? How's it working out?
Absolutely. Thank you. Thank you for this opportunity, to everyone, and Linda for the question. A bit about me. I had a spinal cord injury 9 years ago. So a traumatic, chronic, and originally classified as complete ASIA A spinal cord injury, a motorcycle accident. But before I go into the accident itself, just to talk a little bit about my background. Before the accident, I was selected to become a fighter pilot, to fly the F-16 as a second lieutenant in the United States Air Force. And when this accident happened to me, I was riding my motorcycle home from a regular day of military training, and a van collided with me at 55 miles an hour, resulting in a very severe, like I said, complete injury, where I was in a coma for 3 months, and I was in ICU for 31 days, rotating in a hospital bed from side to side without the intervention to fuse and to address the spinal cord injury, without the ability to address the spinal cord injury because of how unstable I was as far as respiration and all of my other physiological systems. And finally, I woke up 3 months later, February 28th, 2013. And little by little, I quickly saw, thank God I didn't have any cognitive issues, any setbacks of traumatic brain injury related problems. But I quickly was able to use the same principles that I had learned as a second lieutenant in the Air Force, an officer, of observe, orienting, deciding, and acting, and observing and seeing what was around me, what was available, what I could do, and how I, from a patient standpoint, could influence my own neural recovery, my own health, what that looked like, and what success looked like for me 10, 20, 30 years down the road. So that was my initial response to the spinal cord injury. And regardless of waking up from this coma in one of the top 10 rehabilitation hospitals in the US, with the incredible technology, the staff, the equipment, the many possibilities, because I had a complete spinal cord injury, it meant that I would not have the ability to get on these devices and perform and to try and see what could be recovered, or even participate in any gait training program, much less Locomats, or even an FES bike. It was very difficult to get on an FES bike initially. So seeing that from the very beginning, it sent me down this road of wanting to become a researcher myself, first an advocate, to know which way to go, what to do after that binder, after being sent home after a few months and given a binder of how to take care of your bowel, your bladder, your skin, and the secondary issues that we can control. But what's next? Where's the continuum? If you have a complete spinal cord injury, and after inpatient or even outpatient, whatever rehab you can acquire, what's next? And that was the question that I was trying to answer. And how can I, from a patient who's a pilot, an aerospace engineer, nothing to do with the healthcare, how can I, provide my input and influence how this research and my personal neural recovery develops? And the first thing that came into my mind was, well, since I had already gone to Georgia Tech, which is where I'd commissioned through to become a second lieutenant in the Air Force, how can I go back to Georgia Tech and become a biomedical engineer, and learn of ways to bridge the gap between engineering and medicine? And that's exactly what I did. And I graduated in 2018. And from then on, I started traveling all over the US, participating in different clinical studies, again, both as a participant and as a research apprentice, as I'd like to say, of learning of ways that I can optimize and better the results and the responses that I was feeling, and the things that I could communicate to the researchers to better the experiments that were being conducted. And that led me to meeting Dr. Reggie Edgerton, along with many of the top researchers in the field who I've met in different symposiums and conferences, whether it's in Miami at the Miami Project to Cure Paralysis, United to Fight Paralysis, and so forth, many different organizations. And that has been my objective. And that's what led me to move to Los Angeles to start working with Dr. Reggie Edgerton for the last 2 years. Along this process, I also got back in school after moving to Los Angeles, and I'm now halfway through a second master's degree in kinesiology, in human movement. Again, to always try to find ways how I can maximize what I'm doing and what I have control over, which is the effort-driven, activity-based training that's required to make the nervous system activate and the muscles to respond. And what I can say from my personal experience is that this neuromodulation has provided for me an excitability that helps me activate and connect with different parts of my body and what I am sensing. But it's only been when I've become an active and engaged participant of my recovery versus a passive recipient that we started to see the activation of motor control and the transition of me going from Asia B to Asia C. So I'd mentioned that I was initially categorized as a complete Asia A spinal cord injury. Well, for my master's in biomedical engineering at Georgia Tech in 2017 to 2018, one of my experiments that I conducted on myself, since I had access to a Lokomat, was to walk 3 to 4 hours a day for 12 months on a Lokomat with 100% body weight bearing, so 0 body weight support. And after doing that experiment for that 1-year protocol, I was able to then transition my injury from Asia A to Asia B, as I now had new sensations, primarily around my anal region, my bowels. Every time I'd go to the bathroom, I could feel the pressure of the bowels, and when I had to go, and when I had remaining bowels to empty, and that then transitioned to me participating in the neuromodulation clinical trial, and now I'm feeling Asia C responses, which is more sensation from the proprioception that Dr. Edgerton mentioned, but also now being able to control the motor aspect and the contraction of muscles in different movements.
So let me just ask you a few clarifying questions. Now, when you have these sensations, or when you have some functional sensory recovery, does that only occur when you have the device on, or do you have some hangover that you can feel these things even without the device? So you changed to Asia C. So how did you get that way? How do you feel? Do you feel that only when you have the device on, or do you feel it even without the device on?
Great question. Initially, I could say that it was only when I had the device on. Initially, after training and developing a routine and being consistent and activating, like it said, the effort induced side of things. That's when I started feeling that my body was still responding even when the stimulation was off. So once I started feeling that proprioception and that awareness of my nerves essentially started vibrating and tingling, and my brain started recognizing where that tingling and that vibration was occurring. And I would, during the session of me receiving the electrical stimulation, I would essentially try to memorize what that feeling that I would feel during the protocol with the stimulation on while doing the exercises. I would try to memorize what that sensation felt like and how I got to feel that. And I would repeat it over and over and over and over again. That's why I said the key to this is becoming an active and engaged participant versus a passive recipient. And it's at that moment that I started memorizing what that sensation felt like, so that when it was turned off, automatically, not even, I wouldn't even have to try. I would be driving in my car and automatically I would feel my left leg and then my right leg and then my left leg start to activate in a cadence, just like I had done 2-3 hours before that in the protocol. So little by little, that started increasing more and more. And repeating that exercise of meditating also at night when I'm laying in bed, and trying to repeat what I had felt throughout the day in these programs and the exercise. That has been the biggest key, because every time I do that, I start to feel more and more and more. And I'm feeling now as if the effects of the stimulation are lasting longer. Initially, there was just a few minutes, 30 minutes, an hour after the stimulation. Now I can say it's up to a whole day.
And I appreciate your comments about that being active and passive, because I think a lot of the concentration part, the connection between the mind and the body, I think sometimes people just think, oh, okay, I'll do 10 reps of this. I don't even really think about it. Somebody's moving my leg. But if you really concentrate on my legs being lifted up, now my toes are pointed up, now my toes are pointed down. I think all those concepts, that's really huge, but so very often overlooked in the traditional setting, is connecting that mind to that. Now I'm noticing on the little graphic on the screen, the stimulator there looks like it's on the surface over maybe like T12 L1 and then at L2 3. And is that the placement that you use in all your experiments? Have you tried higher? Have you done cervical level? Can it be moved around depending on a person's need? I see lots of head nodding. So good. Who would like
Yes,
question?
absolutely.
So Parag, you want to comment on that?
I'll take that. So the image here is a representation of how the electrodes will be put on the spine. And if you follow the color scheme up there, the yellows over the cervical spinal cord, you can see mapped down to some of the breathing circuits, the hand and arm circuits. Mid-back, which is in the orange, would be where the cardiovascular function would be, the trunk function would be. So it's basically a mapping phenomena. The advantage with the non-invasive transcutaneous approach is the ability to easily move the electrodes up and down the spine and affect all the end organs that are impacted by the spinal cord injury. So unlike an implanted system where you're restricted to a small section of the spine, here we have the flexibility, and depending on the need, depending on what you're training for, depending on what you're trying to target, you can easily move those electrodes up and down the spine and pretty much target the entire spinal cord. So yes, we have done studies where we have done stimulation of the cervical spinal cord to improve hand and arm function, to improve breathing function, over down in the mid-back to improve cardiovascular function, in the lumbosacral area for bladder bowel function, sexual function, standing and stepping, and so on.
And what about autonomic dysreflexia? Has there been any effect on that?
So we closely monitor blood pressure while individuals are receiving the neuromodulation therapy, and we've seen a direct impact positively in how the baseline cardiovascular function is normalized. So we see for individuals that have low blood pressure, we're able to normalize it, and it becomes more closer to what it needs to be. The stimulation so far, touch wood, has never induced an AD episode. So that's really what we're quite happy about. The only time it may occur is when an individual's bladder is full, and it generally subsides the moment a person empties their bladder out. But stimulation itself has never triggered an AD episode.
Wow, that's huge. That's huge. Now, somebody did write in, you mentioned mapping being done, and somebody wrote in, is there extensive mapping? So could you talk just a little bit more about how you do the mapping?
So again, depending on the study that is that's being done, what the focus of the training is. If you're studying training for hand and arm function, we tend to map for individuals to improve hand and arm function. To give you an idea, here's an individual at baseline focusing on hand and arm function versus this after about a month, where they're now successfully able to actually perform these PEG tasks. So this is where mapping comes into picture, where after a few sessions, we're able to identify the right stimulation parameters that are needed to control hand and arm function, versus down in the mid back, where we're stimulating over T11 in this case, where we map for trunk function. So we're slowly increasing the intensity of stimulation, identifying a sweet spot that will work for this individual. So you can see as the stimulation is changing, the person's posture is changing up to a point where it reaches a certain threshold. And we generally use that sub-motor threshold for the training paradigm. So yes, mapping occurs, and that changes from time to time as a person's recovery occurs, their voluntary control improves, they're able to have better control. We have to modify the intensities accordingly.
Which is a great thing. If you have to modify because there's improvement, that would be a great day. There is a question that somebody wrote in, and it's kind of in the same line here. If you have, can you alter bladder function in regards to increasing spasticity? So have you noticed decreases in spasticity because of the training of what's going on in the spinal cord? And then if somebody does have increased spasticity, can you adapt to that? Which I think we've heard the answer, but let's address it specifically.
Great question. So yes, bladder and bowel functions are one of the high priorities for us from a commercial perspective and from a clinical perspective in terms of regulatory approvals. And that's really where our current focus is right now, where when you look at lower unit tract of bladder in general, we're focusing on 4 aspects: incontinence and the ability to store, second, capacity, to have sufficient capacity, sensation, so you know how full your bladder is and when to catheterize, and the eventual ability to voluntarily void. So it's a sort of a complete 360 that we're focusing on from a bladder perspective. So for individuals that have spastic bladder that leads to incontinence and low bladder capacity, in our first study that was done in individuals covering spinal cord injury, MS, stroke, and non-neurogenic bladder, so we have about 35 individuals in this cohort so far, we see about a 70% reduction in incontinence episodes. And that's primarily due to the spastic nature of the bladder. Along with that, we see about a 50 to 80% increase in bladder capacity. So they're actually able to hold more urine in their bladder. And Ignacio can share his experience with this. And more importantly, the ability to actually sense how full their bladder is. So they can then catheterize based on sensation of fullness when convenient, rather than by the clock that, hey, it's 4 hours and it's time for me to catheterize. And the eventual, goal to get to a point where you're voluntarily voiding. Here's an example of an individual with a motor complete SCI at T4. And this is one of the first sessions that we exposed him to the spinal neuromodulation. You can see his legs twitching, and about 10 seconds before voiding, he knows that he's going to actually void. He's got the sensation. And it's hard to see in the video, but there's a nice strong stream of urine that's flowing that's induced by the bladder, the contraction of the bladder and sphincter induced by the spinal neuromodulation. And this is the first time that he was ever exposed. And over the course of weeks of neuromodulation, his thresholds went down, his capacity went higher, his voiding efficiency went higher. So he was emptying more, not just during the stimulation, but also voluntarily. Ignacio, you want to share your thoughts?
Absolutely. I wanted to mention that, and I'll go over the voluntary voiding and the benefits with the bowel and the bladder, but also just in overall, the improvements that I've seen have been in all of the physiological systems of my body. Essentially, when this stimulation is turned on, I feel like my body below my level of injury all of a sudden is turned on as well, and becomes activated, and I start feeling hungry, thirsty, I start sweating, and one of the things that I think I mentioned earlier was particularly when I'm ambulating, when I'm doing gait training using robotic assistive technologies, I start feeling the bottom of my feet every time they touch the ground. So I feel the pressure, and that aids with the proprioception of knowing where my body is in space, and knowing what command to go with next, and how to make the activity-based exercise more efficient, more fluid, and as best as possible. And one of those improvements, like Parag mentioned, has been the bladder function. I have had a suprapubic catheter since 2013, and with the suprapubic catheter, the urine's always flowing out of the bladder. So the bladder capacity, the bladder becomes very atrophied, and the bladder capacity is reduced greatly anytime you have an indwelling catheter, because the normal function of the bladder, expanding and contracting, stops. So urine just goes in and comes right out. So what I've experienced and what I've learned along these years is that leads to many other bladder issues and secondary health complications that can be avoided. And as a participant of this stimulation, this protocol, I've now been able to expand my bladder capacity from around 50 milliliters, which was I originally could hold in my bladder, to now around 250 to 300 milliliters in my bladder. Apart from that, that suprapubic catheter, in order for me to not have the urine just flowing continuously, in these 9 years that I've had the spinal cord injury, I developed a strategy for myself of buying a clip, being able to clip the suprapubic catheter. Sometimes people put a plug at the end, which is something very similar, to be able to control when the urine comes out and when it's inside the bladder. And by doing that exercise of letting the bladder expand and contract and only opening the valve or the clip when I have to void, that has allowed me to train my bladder and that sensation. So originally I would feel this when I had to go. I would feel this chill, these goosebumps, like the beginning of what's called the autonomic dysreflexia. But it would happen just momentarily and very quickly for about just a few seconds. So I wouldn't call it full on AD, but I would feel the chills, and I would feel this sensation that my bladder was full at the beginning. But now, as a participant of this trial, I'm not feeling that nasty sensation, those goosebumps, the sweating that I'd get when my bladder was full, that would then indicate for me to open the valve and open the clip, and then that sensation would immediately go away within seconds. I don't get that any longer. I get a sensation that's different. It's different. All sensation is very different than what it was before the accident. But I get a sensation that's different than what I had before the neuromodulation trial. And I'm now able to control how much, when to open the clip or when to open the valve. Because before that, whenever I'd get that sensation, if I didn't open the valve or the clip within 5 to 10 seconds of having that sensation, I would immediately start voiding through my urethra and have an accident on myself. So I would always have to open the clip immediately. But now, for example, I could be driving and I'll get this sensation that is not the autonomic dysreflexic type of feeling, and I can hold it. I can control it till I can pull on the side of the road or get to my destination. I've been able to hold it 5-10 minutes with that sensation, and I haven't peed on myself. So that has been so transformational in my care for my bladder. Also, my bowel programs have gone down from an hour to 20 minutes, 20 to 30 minutes max. 25 minutes, I'd say, is a good average of going to the bowels, feeling when I have to go, how much I have to go, and you know, utilizing strategies like putting a vibrating massager on my stomach while I'm sitting over the toilet, going to the bathroom, letting gravity do its thing. This massager that's vibrating on my stomach, on my intestines, let that massager help a little bit, or so I think. But also, more than anything, that proprioception, that sensation of that the neural stimulation has brought about, has been the key factor of me being able to tell, okay, I think I'm done, or no, I feel like I still have to go. Like, perfect example was this morning. And having those sensations is really, really helping me with my care and my bowel and my bladders.
So that leads us into a question that somebody wrote in about, are you having any pain? And I'll differentiate that question just a little bit. Do you have pain when you have just from the device? Is there any pain? A lot of people will want to know that. Do you have sensations, these AD sensations or different kinds of feelings? Or do you ever have pain? So like if you had your foot caught in the wheel of your wheelchair or something, would you feel the pain from that?
Well, yes and no. I want to highlight that the sensation is different, is very different. And I don't know if it'll ever get to the point where it's exactly to what it was before the accident, or to the sensations that we feel above the level of injury. So if my foot, to follow your example, if my foot is caught on something and it's going through pain, obviously I don't feel the pain like before the injury, but I do feel that something is painful. Something painful is happening to my foot, and that's translating by how I am interpreting the sensation. For example, when the bottom of my feet are touched, I start feeling the tingling and the sensation in my chest, in my wall of my chest, and my thorax, and in my back. And for example, if I'm touched on my left foot, the left side of my chest starts tingling. If I'm touched on the right foot, the right side of my chest, and that's how I'm able to differentiate. But the pain is interpreted by sensations of chills, like I said, but it's hard to describe it because it's not just chills it's not the same chills of AD, and it's not the same chills of my bladder is full. It's a different type of sensation, but I can and I do recognize when I have pain. Now, the neurostimulator itself does not cause pain. The only pain that I have in my body is from an extreme neuropathic pain caused by a secondary injury, which is a brachial plexus injury, where I ripped the peripheral nerves of my right arm in the motorcycle accident from my spinal cord. So that's separate. And the spinal cord, the neurostimulation is actually helping that, helping tremendously and significantly reduce the neuropathic pain in so many ways that both meditation and medication have never been able to. So that's my experience with that.
So here is the really big question. When will this be available to individuals out in the community? Can people participate in the studies? What are the next steps for bringing this to people who need this kind of therapy?
So this is currently undergoing clinical trial right now. We have our first multi-center clinical trial ongoing at about 7 or 8 different clinical sites around the US. And this is focused, the focus of the trial is neurogenic bladder. So individuals that have neurogenic bladder due to a stroke, spinal cord injury, or multiple sclerosis are eligible to participate in this trial. And our hope, our expectation is by the end of this year, we'll have the trial completed. And our intent is to make this device available as early as next year to individuals with neurogenic bladder. And we are actively pursuing getting this on the market as soon as possible.
And I'm sure there's a lot of listeners who are very eager to get this particular device. Will this be for adults only? Multiple diagnosis? Will this be for children, or will you have an alternative study going on for children?
Correct. So this initially is focused for adults. We have a separate device that is being built for children, pediatrics, and that's what we call a SCIP, what you see on the right side here. So that is being developed right now, and we're hoping that SCIP will be brought to market in sometime 2024 with a different clinical trial, since it's for pediatrics and in a different indication. So SCONE, our flagship device, will be for adults and be available next year.
Okay, and when it is, I hope you let us know so that we can announce it on the Reeve Foundation, because people will want to know. I'm sure they can follow SpineX webpage. Do you have a website that they could follow along?
Yes, absolutely. So it's spinex.co right here on the bottom. So you can follow us on our webpage or on social media, where we do announce our status and any new updates that come out.
Okay, now there's one more question. It's kind of a little bit aside from this particular device, but maybe it has something to do with it. But there's a question for Dr. Edgerton about neural stem cells and 3D neural matrix use. Any final thoughts that you have on that, on those particular notions?
Well, I think one of the advantages also of this approach is that I think it has potential to be combined with other types of interventions. A number of people have consulted with us about the feasibility of combining the stimulation with stem cell implants and other types of methods to stimulate regeneration. And I think it's inevitable almost that that's probably going to occur sometime in the future. But it's too early now. I mean, we don't know enough about either one of these interventions to start human trials in that right now.
Well, but it is on the table. So that is, research begets more research. One final question is this for lumbar injuries as well as for higher level injuries? And I think you've alluded to that.
Yeah, certainly. This is one of the first things that we wanted to try as soon as we saw the results with the epidural stimulation and how we thought it was working. And we still think it's working just like we thought it was after that first subject. But we wanted to test it to see if it was beyond the concept, for example, that most people think about central pattern generation. It does not depend on the classic concept of central pattern generation as is usually thought of. So it works. It seems to be working in autonomic neural control, sensory motor control. This is one of the exciting things about it. The mechanisms, there's some very central mechanism, fundamental mechanism throughout our physiology that's allowing us to increase the plasticity of these neural networks so that then we can instruct these neural networks how to reorganize through training activity dependent mechanisms. So it's has lots of potential to be combined with many other interventions.
So as wonderful as this is, it sounds like you're just scratching the beginning here. Although this is a pretty good as it is, but still lots more thought to go into this, lots of continued work. And we all appreciate that from the community, as well as we appreciate all three of you participating today. This has been a wonderful opportunity I have enjoyed hearing about the latest and greatest and what's going on, and where it certainly brings a lot of hope for the future.
Well, let me make one other comment relative to what you just said. I often use at the end of my lecture a photograph of the Wright brothers flying. And that's about where we are right now in terms of realizing the potential of recovery of function.
That is very much a great idea. That really sums it up. So a lot more to come. Kind of rudimentary right now, but still it offers so much to so many. So we appreciate all three of you being on today. We appreciate your efforts for everything you're doing. This offers a great deal of hope, and we just thank you so much.
Thank you.
Thank you.
Pat. Yes.